P. Aruna, lecturer in microbiology from YA Government Degree College for Women, Chirala, Prakasam district. In this session, you are going to study or you are going to know about information about antibodies, basic introduction and types of antibodies, and finally applications of antibodies in various areas. So, let us first start with the antibodies. So, antibody is nothing but a large Y-shaped glycoproteins formed by plasma cells in response to antigenic stimulus. So, when a foreign particle or an antigen enters into the body, the immune system responds and produces and starts the production of antibodies. These antibodies are produced by B cells of the immune system. It recognizes the unique molecule of the pathogen called antigen. So, the antibodies, they show the immune response and immediately they bind the foreign particle which enters into the body and induces the response. Coming to the structure of antibodies. First, the structure of the antibodies was revealed by the persons called Gerald Edelman and Rodney Robert Porter in the year 1952. So, according to their structure, according to their analysis, the antibody is made up of 4 polypeptide chains. They are called as light chains and heavy chains. The light chains are there are 2 light chains which are identical, and there are heavy chains which are again 2 in number. They are called as heavy polypeptide chains. So, they have given this structural analysis in the year 1952 itself. So, according to their analysis, the antibody is made up of 4 polypeptide chains. Here you can see in the structure, you can see on the screen the region of the antibodies. These chains are bonded by interchain disulphide linkage. Light chains are of again 2 types, they are called as Kappa chain and Lamda chain. However, each antibody may contain any of the 2 chains, either it contains a Kappa chain or it contains a Lamda chain. These 4 linked polypeptide chains are arranged in a flexible manner maintained by the hinge region, where you can see here on the screen, this is the hinge region which gives the flexible nature to this antibody. So, the antibody is having a capacity or it is capable of binding to the antigens which are maybe bivalent or multivalent. So, this hinge region plays a major role giving the flexibility to the antibody. Again, when come to the light chain, it has 2 successive domains called as constant domain and a variable domain. So, these 2 domains, they help in maintaining the structure of the antibody or help in maintaining the antigen to bind to the antigen. Each heavy chain has 1 variable region with a variable domain and a constant region with 3 constant domains. The antibodies has 2 functional regions called FAB region or FC region. FAB region is called as fragment antigen binding region. So, this region helps the antibody to react with the antigenic determinants at antigen binding sites. The antigen binding sites are nothing but the epitopes present on the antigen to which the antibody binds. So, this FAB region, that is a crystalline fragment antigen binding region, helps to bind with the antigenic determinants present at the antigen binding sites. Here you can see the structure on the screen. So, this is the FAB region and this is the FC region. So, this region will help to bind the antibody to the antigenic determinant site, that is epitope. Here you can see the variable region or heavy chains in the constant region. This is a hinge region. Here the chains are binded by disulphide linkage. Coming to the second region, FC region, which is a functional region called as a fragment crystallizable region. It is the site at which the antibody molecule can bind to a cell. So, this site will help the antibody to bind to a antigen. It enables that each antibody generates an appropriate immune response for a given antigen. So, whenever an antigen enters, so the response, the proper response should be given is indicated by this FC region only. Next, coming to the functional sites present on another functional sites present on the antibody, that is paratope. It is an antigen binding site of the antibody which is present at the FC region. So, paratope is nothing but a region present in the antibody which binds to the epitope of the antigen. So, this paratope is present in the antibody. Next region is an important region called hypervariable region. This region itself contributes to specificity of each antibody. So, generally all antibodies are having similar structures, but these number of antibodies can be shown a little variance at a particular region present at the tip of the chain, that is nothing but the hypervariable region. So, sometimes the folding at the hinge region may take place. So, while folding this hypervariable regions, they come together and forms the antigen binding pockets. So, these pockets are the sites that are in close contact between antibody and antigens. So, they help in maintaining the close contact, and those particular sites are called as complementary determining regions of the antibody. So, these antibodies binds to specific antigens by hydrogen bonds, ionic bonds, hydrophobic interactions and van der Waals interactions. So, these are the interactions they can use while binding to the particular antigen. Next, coming to the different types of antibodies and the terminology given to the different antibodies. Allo antibodies, they are produced after exposure to the non-self antigens of the same species, that is they belongs to the same species, but they do not belong to the same. So, they are called as non-self antigens. These non-self the antibodies which respond to these non-self antigens are called as allo antigens. Second one is auto antibodies. These auto antibodies are produced due to the exposure to the self antigens. Next, coming to the functions of the antibodies. Let us have a glance to the various functions of the antibodies they perform. So, neutralization. Neutralization is nothing but it is a process in which the foreign substance or an antigen is totally inactivated and it is made non-functional. The second one is opsonization. Opsonization is a process is nothing but targeting the particles for destruction through phagocytosis. Here the thing which is involved is called as opsonin, and the final product is nothing but the phagocytosed product which is totally inactive. So, the targeting the particles for destruction by phagocytosis is called as opsonization. Next one is activates the complement system. So, the complement system is activated and the destruction of the bacterial cells is taken place by lysis. The last function is inflammation. Inflammation is nothing but the body's response for the injury. Whenever an injury takes place, the body, the internal immune system responds and induces a set of reactions. These set of reactions finally they help in destroying the pathogen. So, in this session, in this topic, we have learned about the structure of the antibody, how it functions, the parts or the functionable areas of the antibody and various functions performed by the antibodies. So, we have learnt about structure of antibody. Now, coming to the different types of antibodies. Before entering into the or before going to explain about the types of antibodies, antibodies are otherwise called as immunoglobulins. Antibodies are otherwise called as immunoglobulins. So, let us study the various types of immunoglobulins. So, the heavy chains are of again 5 types, that is gamma, alpha, mu, delta and epsilon, according to which the immunoglobulins are divided into 5 classes. That is nothing but immunoglobulin G, immunoglobulin A, M, D and E. So, coming to the first immunoglobulin, immunoglobulin G. This is the most abundant immunoglobulin among the immunoglobulin pool, which is accounting to about 80% of the total immunoglobulin pool. So, it is a bivalent antibody. It has a half life of 7 to 23 days. It is a molecular weight of 1,50,000 Daltons. Again, as it is a bivalent antibody, it is having again a heavy chain and a light chain. Heavy chain is made up of composed of 440 amino acids and light chain is composed of 220 amino acids. Here you can see the structure of this immunoglobulin G. This is the variable region, this is the constant region. I have already told to you that the main difference varies to each antibody is in this variable region itself at this tip, which is having the hypervariable regions, and this is the heavy chain and the light chain. So, this is the structure of the immunoglobulin G, and it is called as a maternal antibody because this antibody can cross the placenta and it produces immune response to the newborn. These immunoglobulins are divided into 4 subclasses: IgG1, IgG2, G3 and G4. So, these 4 types of immunoglobulins subclasses, they are performing different types of functions. The crystallizable fragment portion of immunoglobulin G1 and G2, these two bind to the macrophages and neutrophils, and finally, they destroy the pathogen. Again, the crystallizable fragment portion of immunoglobulin G1 and G4 helps them to cross the placenta and provides the immune response to the newborn. Here you can see the structures of different types of immunoglobulins, immunoglobulin G, 4 subclasses G1, G2, G3 and G4. Coming to the biological properties of this immunoglobulin G. So, we have already known that it is the only immunoglobulin that crosses the human placenta and offers a passive protection or passive immunity to the newborn. And this immunoglobulin G is having the property to activate the classical complement pathway during antigen antibody reactions. So, activation of the classical complement pathway leads to the production of various components which finally kills or destroys the antigen. Next, this immunoglobulin G can neutralizes the toxins and viruses. So, this is the one of the important immunoglobulin which can neutralize the viruses. So, by binding to bacteria, this immunoglobulin G opsonizes them by the process of opsonization and enhances the process of phagocytosis. So, when the process of phagocytosis is enhanced, the phagocytic components or the stepwise phagocytosis process will take place and finally, the pathogen is killed. Coming to the second class of the immunoglobulin, this is immunoglobulin A. This is the second most abundant immunoglobulin in humans. It occurs in saliva, tears, breast milk, colostrum and the mucosal secretions. This IgA is observed mainly in body secretions and protects the internal body surface. So, it is termed as the antiseptic paint of the mucosal membrane, or also it acts as a paint on the mucosal surfaces. Here you can see the structure of the immunoglobulin A. This is also called as secretory immunoglobulin. Here the crystallizable fragment portion of the immunoglobulin A binds to the components of the mucus and contributes to the ability of mucus to trap microbes. So, it is a I have already told you that is also called as a mucosal paint. Here you can see in the structure the secretory part of the immunoglobulin. It binds to the antigens and especially it is present in the mucous membranes and traps the microbes in this part. Coming to the biological properties of immunoglobulin A. This immunoglobulin plays a considerable role in the generating the alternative complement pathway. So, it is also produces immunity against tapeworms which are present in the intestine, and it is also highly present in the colostrum. So, when the baby is fed with the breast milk, it gives a protection from the intestinal pathogens as it is present in the colostrum of the milk. The third type of the immunoglobulin, third class of the immunoglobulin is immunoglobulin M. It is also largest antibody and third most antibody present in the human serum. It has a half-life of 5 days. It is a polymer of 5 monomeric units, each monomeric unit possessing 2 heavy chains and 2 light chains. So, as it is having each monomer is having 2 heavy chains and 2 light chains. So, it is having 10 epitope binding sites. I have already told you that epitope is the site of the antigen where the antibody binds. So, here you can see the structure of this immunoglobulin. It has 5 monomers. They are arranged in a pinwheel array. These 5 monomeric units are held together by disulphide bonds and are linked by a special chain called as J chain. Here you can see in the structure, this is the J chain. So, these 5 units, monomeric units are arranged in a pinwheel array manner, and this is one of the giant immunoglobulin among the other types of immunoglobulins. Coming to the biological properties of this immunoglobulin M. It is the first antibody to appear in the primary response. So, whenever the antigen enters into the body, the immune cell shows its response in a stepwise manner, that is primary response, secondary response, as such like that. So, it is the first antibody, it appears or it produces during the primary response itself. So, when this levels of the IgM is detected in the newborn baby, it gives an useful indication that the baby is suffering from an intrauterine infection such as congenital syphilis, rubella, toxoplasmosis, etc. So, whenever newborn is detected with this type of immunoglobulin M antibodies, it gives an indication that the newborn is suffering with certain diseases, certain infections. Next, this IgM shows a high functional affinity for multivalent antigen. In the structure itself, in the previous slide you have seen the structure. So, as it is having 10 epitope binding sites, it is having the ability to bind to the multivalent antigens. It also shows the properties such as opsonization, complement fixation, agglutination, cytolysis, etc. The next class of the immunoglobulin is immunoglobulin D. It occurs in very low concentration in the blood serum, that is only 0.2% of the total serum antibody pool. It is a monomer and has 2 epitope binding sites. It has a half-life of only 3 days, and this immunoglobulin is found on the surface of the B lymphocytes and acts as a receptor for antigens. It is abundant in memory cells and it is play an important role in secondary immune response. So, primary response, we have already seen that immunoglobulin M is the first antibody which responds during primary response itself, whereas immunoglobulin D, it responds during the secondary immune response. Here you can see the structure of the immunoglobulin D. Here is the antigen binding site and the variable part of the chain and the constant part of the immunoglobulin. Biological properties. Immunoglobulin D is associated with the surface of B lymphocytes along with IgM, is found to act as an antigen receptor. So, these two along it alone cannot bind. So, in association with IgM, it acts as a receptor, antigen receptor, and binds to the antigens, and it plays a major role in generating self-reactive autoantibodies. So, it has the capacity to generate the self-reactive autoantibodies. We have already seen in the first session that difference between autoantibodies and alloantibodies. So, it is having the capacity to generate the self-reactive autoantibodies. Next, coming to the immunoglobulin E. So, it occurs in blood serum in extremely low amounts, very low amount among the immunoglobulin pool. It has half-life of 2 days and with a molecular weight of 1,90,000 Daltons. It is a monomer and has 2 epitope binding sites. The production of this immunoglobulin is in response to parasitic worms and also in response to allergens. Whenever any allergen enters into the body, immediately the immunoglobulin E production takes place and it responds to that particular allergen. Here you can see the structure of this immunoglobulin E, same FAB region, FC region, heavy chain, light chain, variable region and constant region. How the let us see the mechanism, how it inactivates the allergen. So, it binds to the crystallizable fragment receptors on the membrane of basophils and tissue mast cells. So, when it binds to the mast cell, when the allergen along with this when an allergen enters into the body, here the antibody, it binds to the mast cell, and by the cross linking of the these cells, it induces an immune response by degranulating the mast cell. So, the degranulation of this mast cell leads to the vasodilation or causes dilation of the local blood vessels and resulting in allergic reactions. So, here you get the mechanism of the mast cell degranulation in this picture. So, the cells degranulate when 2 IgE molecules are cross-linked by binding to the same antigen. The vasodilation of the blood vessels will take place, dilation of the blood vessels will take place, resulted in the production of different products such as heparin, vasodilated products are released. Biological properties. The high levels of IgE with worm infestation, they plays a protective role, that is when heavy infection takes place in the intestine, high levels of IgE is secreted in the body and they play a protective role. So, also IgE mediates the reagenic hypersensitivity. So, different types of hypersensitivity, type 1, type 2, type 3, type 4 hypersensitivity reactions were there. So, IgE mediates the type 4 hypersensitivity reaction. So, in this session we have learnt about the different classes of the immunoglobulins, their structure and the various functions they perform during an immune response. we have already seen the structure of the antibody, different classes of the immunoglobulins, and let us focus on applications of immunoglobulins. We have already seen in the previous slides about the various properties of the immunoglobulins. Coming to the applications in a diagnostic field. Increased concentration of immunoglobulins is an indicator in diagnosis of many diseases. That is, example, increased levels of immunoglobulin M is an indication of viral hepatitis. So, not only M, when an increased levels of the various types of immunoglobulins is observed in the sample, it indicates that various disease conditions in the body. So, it is used in most immunodiagnostic techniques, especially ELISA is nothing but a immunoglobulin assay which uses multiple antibodies to detect specific antigens capable of causing infectious diseases. So, ELISA technique is used to study the antigens or to know or to detect the specific antigens which are causing specific different types of infectious diseases. So, increased levels of immunoglobulins in the serum helpful in analyzing a patient's antibody profile. So, whenever the immunoglobulin level increases in the patient's serum, it definitely helpful in analyzing the patient's antibody profile. So, antibodies are also used in pregnancy detection test kits, in which the antibodies binds to the human chorionic gonadotrophic hormone. So, this HCG or human chorionic gonadotrophic hormone is a hormone secreted by the placenta during pregnancy. It can be detected in the early days of the conceiving time itself, that is within 9 days, by using this pregnancy detection kit, in which the antibodies are given, multiple antibodies are given, which can react with this human chorionic gonadotrophic hormone and gives a visible test result, so that pregnancy can be confirmed. This is these immunoglobulins are used to treat hypogammaglobulinemia. So, hypogammaglobulinemia is a condition in which a sufficient amount of immunoglobulins are not produced due to some defect in the immune system. So, when sufficient amount of immunoglobulins are not produced, then the body is unable to respond to different types of infections. It is a defect. So, by giving ready-made antibodies to these affected persons, it gives a passive immunity to the person. That is, ready-made antibodies are administered to the affected person to overcome this hypergammaglobulinemia to induce a passive immunity. Next application is Western blotting. It is a blotting technique in which the labeled antibodies are used to detect the protein, antigenic protein. This blotting technique is a very helpful technique in identifying the protein nature of the antigen. The use of immunoglobulins with HIV infected children was associated with lower rates of bacterial infections. That is, when the child is infected with HIV, then the immunoglobulin combination is given to that infected person. It gives the lower rates of bacterial infection, that is, the child is able to overcome some of the bacterial infections. Immunoglobulins used to treat antibody mediated inflammatory and hypersensitivity diseases. So, various hypersensitivity diseases you can see myasthenia gravis, arthritis and different types of diseases, in which the immunoglobulins are used to treat those hypersensitivity diseases and also various inflammatory reactions. Next one is it's an antibody dependent cell mediated cytotoxicity. target organism is coated with antibodies and they are bombarded with chemicals from non-specific immune cells. So, ADCC is induced by the immunoglobulins, so the antibodies which are coated with the antibodies and they are bombarded with the chemicals from non-specific immune cells. So, these are the few applications in the diagnostic field. So, antibodies application is in every field, it is most helpful in identifying the pathogenic proteins or various pathogens. In this session, hope you understood all the about the basic structure of the antibody, classes of antibodies or immunoglobulins, and especially functions related to the diagnostic field.
टारगेट में